Okay, I think we might as well go ahead and get started. My name's Ron Oldfield. I'm the host for today's biology department seminar and also Year of Darwin special event. And for today's event, we are having Dr. Thomas Neer from Yale University give a seminar on climate change and its effect on Antarctic fishes. It's really a pleasure to introduce Dr. Neer. He's had a very impressive history. He did a Ph.D. at the University of Illinois and then went on to do a postdoc at University of California, Davis. And after that, got a position as an assistant professor and I think also a curator of fishes at the University of Tennessee. He had an academic appointment and also was a curator in the museum there and worked there for 3 years and then launched into a position as assistant professor of ecology and evolutionary biology and also a curator of fishes in the Peabody Museum of Natural History at Yale University. And he's been there for a couple of years now doing research and teaching biology of fishes. Dr. Neer's research is very interesting. He takes molecular data from fishes, he extracts the DNA and looks at sequences and looks at closely related species and puts the molecular data into computer programs that then give him the proposed or hypothesized evolutionary history of the group. And with these types of data sets, he does things like calibrate the history of the group with fossils and look at the rates of molecular evolution in different lineages. He's able to test different historical biogeographic hypotheses, basically trying to determine when certain species were in certain areas and how they went from one area to another. I've also seen him do studies that use these hypotheses to test for key innovations that may, or evolutionary novelties that may have led to the diversification of different lineages. These are all very interesting, and my feeling is that this will probably touch on one of those in today's talk. Yeah. So without any more ado, without further ado, let's move it on to Dr. Neer. Thank you.
Thank you.
Oh, and one more announcement. We are going to dinner after the seminar this evening, so if we have room for one more person. So if anyone's interested, please talk to Mike Bernard after the conclusion of the seminar, and we can make arrangements to all meet for dinner. All right, thank you.
Thank you for that introduction, Ron, and thank you everybody for hosting me for this visit where I have an opportunity to come share my research. I'm going to try to figure out the lights here, just because I think we will be able to see some of the images perhaps a little bit better. There we go. Okay, so as Ron pretty much touched on a lot of my main research themes. I'm a phylogeneticist. I'm also an ichthyologist. I study biodiversity of fish. And I'm really interested in using phylogenies of fish to test, long-standing, in fact, develop new hypotheses about how macroevolutionary trends result in the biodiversity that we see on the planet. And in many ways, I think we could get at several of these questions by studying this bizarre assemblage of fishes that occur in Antarctica. And in fact, I think it's safe to say that Antarctic fishes really are the only example of adaptive radiation in marine fishes. Perhaps one example are the Sebastes rockfishes along the Pacific coast of North America that kind of dribble over to the western side of the Pacific as well. But I think it's pretty clear that what we see, the remarkable ecological and morphological diversity we see in this group of closely related species, is essentially unrivaled among other groups of marine fishes. And that just to have a line of caution here, just go back to Samuel Clemens. He said, don't tell fish stories where the people know you. Unfortunately, some of you do know me. But particularly don't tell them where they know the fish. So if nobody here is familiar with Antarctic Notothenioids, perhaps today we'll find ourselves in good company. So my questions are going to be rather basic, perhaps. So I'm going to try to tell you a story. The story I'm going to try to tell you is why Antarctica is such a unique marine habitat that's essentially unlike any other marine habitat that we have on the planet today. And then also I'm going to try to build up this story of this story of an evolutionary diversification of a really small piece of the tree of life of fish, and how this small piece of the tree of life of fish can give us really interesting insights into these patterns of adaptive radiation. So the first question is rather basic, asking what are the phylogenetic relationships? And by phylogenetic relationships, I mean those ancestor-descendant relationships that are analogous to a family tree. So we all have a family lineage. The difference is our family lineages reticulate. Mom and dad come together, and we are the product. But with phylogeny, you could think about it, it's analogous, but kind of turn it on its head, where we think of lineages diversify, okay? So it's kind of in a sense, a little bit of a backwards way of thinking of it, but it's very analogous to a family tree. So you'll see a lot of these diagrams today. And these diagrams essentially are these branching dendrograms where the species that we're studying are out at the tips. So basically what we're arguing when we look at a phylogeny is that these species are more closely related to each other than either is to species over here. Okay, so we're reconstructing these branching diagrams. And then I'm also interested in asking, are there broad biogeographic patterns with the diversity of this particular group of fish that I'll be talking about? These are called the Notothenioids, and I'm sorry there's no common name for them, but if anyone has ever eaten Chilean sea bass, you're familiar with Notothenioids. And what we see is that Notothenioids have this really interesting biogeographic pattern to where several species and major lineages are distributed to peri-Antarctic areas such as southern South America, southern New Zealand, southern Australia, southern Tasmania. But then the bulk of the biodiversity of this group of fish called Notothenioids are right here in the Southern Ocean around the Antarctic continent. So we're interested in asking, does this really make sense? Is this just like a jumbled pattern, or is there a pattern with the biogeography that matches the phylogeny? And then ultimately, what I'm interested in the story trying to tell you today is to ask, when we look at the phylogeny of these fishes, so here's actually a phylogeny of penguins. And as Ron alluded to, what we can often do when we have a phylogeny that's based on genetic data, such as DNA sequences, we can attempt to convert those branches, which we estimate as the substitutions per site, that is per stretch of DNA we're looking at, try to convert that to some absolute age estimate. And so what we have here is a phylogeny where now the branches are drawn to correspond to the actual estimate of evolutionary time. And what we see here is a phylogeny of penguins, and what we see here is a hypothesis of the paleoclimate of the Southern Ocean. And what these authors are arguing is that during different periods of penguin evolution, as climate change happened in Antarctica, we've seen resulting patterns in the penguin phylogeny. So I'm interested in asking that with the Notothenioids, that do we see a pattern in the timing of their evolution that correlates to the pattern of the timing of climate change in the Southern Ocean? So what I'm going to do today in terms of background, I'm going to tell you a little bit about the Southern Ocean. I'm going to tell you a little bit about the diversity of Notothenioid fishes. There's approximately 125 species. I'm going to argue that they indeed are an example of an adaptive radiation a priori. And then we'll talk a little bit about this hypothesized key innovation, this antifreeze glycoprotein that all the Antarctic Notothenioids possess. So first about the Southern Ocean. The Southern Ocean was the ocean I had never heard of before I started my study on Notothenioid fishes, and I began this research when I was a senior graduate student. So I felt I was pretty disappointed I didn't know about the Southern Ocean. The Southern Ocean is interesting in that essentially, if we kind of look at the planet from the bottom up, what we see is that Antarctica is essentially a continent that's surrounded by an ocean, that is the Southern Ocean. Whereas if we think about the Arctic essentially is almost, you could think of it as an inland sea that's surrounded by continents. And that has very appreciable consequences with regard to seasonality of winter versus summer, and also long-term paleoclimate. Then also, this is the main thing that one needs to learn about the Southern Ocean with thinking about Antarctic biogeography as well as Antarctic climate, paleoclimate, is the fact that really the only circumpolar current on the planet is the west wind drift, or the Antarctic circumpolar current. And basically, it's a current that goes around and around in a circle that surrounds Antarctica. Now, when this current started is an area of debate, and it has to do when southern South America and the Antarctic continent finally split. I'll talk a little bit about that. But what's essentially happened with this circulating current, it's essentially prevented any warm water currents from reaching the continent. So as Antarctica began getting cold, it just kept getting colder and colder and colder because no warm water could come in and actually bathe the coast. So I grew up in Chicago, and the winters really were bad, right? Now I live in New Haven, Connecticut, and the winters are not that bad. And so I can thank the Caribbean and the currents coming off of Cuba for that. Antarctica doesn't have that luxury that we have in the Northeast. Now, with regard to the fish biodiversity in the Antarctic, the fish biodiversity in the Antarctic is unlike any other marine habitat on the planet. That is, if we went to any marine habitat, including the Arctic, and we were good ichthyologists and we sampled all the available habitats, we would find representatives of all the major clades or the major lineages in the fish tree of life. However, in Antarctica, what we find is a really limited sampling of the biodiversity of fishes. And I'm going to tell you why later in the talk. So if we look at the diversity of fish, 75% of all the species are in this one group, or clade, meaning a natural lineage, the Notothenioids. In terms of abundance, about 90% of all the species that you would go out and catch are from this one little clade, or one little lineage. And then biomass, that is if we actually weighed all the fish we caught, again, it's rather similar to abundance. About 90% of the biomass are individuals that are from this one little puny clade in the fish tree of life. And this is a situation that we see in no other marine habitat on the planet. No marine region of the world, I should say, not habitat. So as I said, this circumantarctic current has created this thermocline in sea surface temperature that's known as the Antarctic polar front. That is, there's an appreciable difference in the temperature above and below this front. And this is a very strong barrier, thought to be a barrier both to dispersal to the Southern Ocean and dispersal out of the Southern Ocean, all right? And not only that, this is characterized by very deep sea habitat. So there's no continental shelf habitat that would connect the Antarctic Peninsula with southern South America. So not only is there this open water habitat barrier, but there's a real strong thermocline in temperature as you cross this Antarctic polar front. Now, the story of the Southern Ocean and the story of Antarctica is ultimately tied into a paleogeographic story of the former super southern continental landmass Gondwana. And most of you probably know that the reason why a lot of organisms in Africa and South America are very similar to each other, plants, animals across the tree of life, is because Africa and South America used to be connected about 95 million years ago. And so what happened is that Antarctica here is a player in this story of the fragmentation of Gondwana. So not only was Antarctica in a more temperate latitude at one time, but actually had physical connections with other continents that are still with us, such as South America and Australia. And if we look at the timing of this paleogeography, what we essentially see is that by 65 million years ago, Antarctica is becoming isolated from these other southern continents and moving to a more polar position. And what we see when we look at, here's the paleotemperature estimate. These are from oxygen isotope ratios in diatoms, I believe. And what we see is that there's a sharp change in temperature, where temperature becomes drastically cold right at this Eocene-Oligocene boundary approximately 35 million years ago. This is a worldwide pattern in climate change. And we see it particularly in the Southern Ocean. And here we start seeing the appearance of Antarctic ice sheets at about 35 million years ago. So the story is, as Antarctica is separating from these continents, it's creating this opening here, the Drake Passage, that allows that circumantarctic current to form, that then facilitates this essentially development of polar conditions in Antarctica. And this is a branching diagram that relates how we think those continental areas were connected. So this is only analogous to a phylogeny. And so what we see here is essentially these are all of the land masses that are part of Gondwana. And what we can see that the last connection that Antarctica had to basically New Zealand and New Caledonia and Australia, because here's Antarctica here, is about 80 million years ago. So we want to think about 80 million years as kind of this initial fragmentation of Antarctica from Gondwana, and then the hypothesized date of split between that southern South America and the Antarctic Peninsula was 30 million years. However, isotope ratios from fossil fish teeth have shown that essentially there's a strong signal of mixing between Pacific and Atlantic waters around southern South America and the Antarctic Peninsula. And so now we're pushing the date back to that opening of the Drake Passage to about 42 million years ago. So what we're seeing is that as Antarctica is becoming more and more isolated, allowing the Drake Passage to form, maybe approximately 40 million years ago, then we're seeing this signal of climate change where basically the world as a whole, but Antarctica in particular, is getting very cold at about 35 million years ago. And this is exactly what we see. If we look at benthic foraminifera, its oxygen and carbon isotope ratios, we see this really strong signal of climate change where as we're approaching the present day and we hit at about 35 million years ago, we have this marked change in these isotopes, which are then inferred as a marked change in the actual temperature, getting colder. Now, what do we know about the fishes that were around in Antarctica before it got cold? I'm telling you that Antarctica is a unique marine area of the world and that it is essentially biodiverse, has depauperate biodiversity. Well, we have this fascinating fossil formation at the tip of the Antarctic Peninsula, here on Seymour Island, the La Meseta Formation. That's anywhere from, I think, stretches from 55 to about 38 million years ago. And what we see is that when we look at the fossil fishes in this formation, this is what we would catch off the coast of Connecticut. This is a rather cosmopolitan, temperate, I almost said freshwater, a cosmopolitan, temperate fish fauna. And in fact, you know, really neat stuff like billfishes, cutlass fishes, even wrasses, which are kind of your poster child of coral reefs, as well as a whole host of chimeras, sharks, skates, and rays. So if we look now what's around after the present day, essentially all of these groups are gone. The only groups that are around are skates and these Notothenioids, okay? So what we see is this marked change from a temperate marine cosmopolitan fauna to one that's rather restricted. And we know that because we've gone out and we sampled all those fishes like good ichthyologists, and we've seen that there's very little representatives of these other groups of fish. Now, we're fortunate to have a nice fossil, a preserved neurocranium of a fossil species that is thought to be closely related to this guy. And this guy is actually one of those weird Notothenioids that lives outside of the Antarctic. It's temperate. It's found in southern South America and the Falkland Islands. And so what we know is that this critter, or that lineage, has been around for at least 40 million years because we have a fossil that is closely related to this guy. And I'll talk more specifically about that when we talk about converting molecular trees to absolute divergence times. So this fossil is from that formation that we've learned so much about, that pre-getting cold fish fauna of Antarctica. Okay, let's talk a little bit about the Notothenioid diversity. And I love this chart. I kind of stole this chart from a friend of mine named Joe Eastman, who actually teaches at Ohio University. And if we look on the y-axis the number of species and look at the year that the species was described, what we see is that at a time of the height of polar Antarctic exploration is where we have this really high rate of species description. So zoologists were going down there catching fishes, putting names on them, you know, freaking out because they found fishes whose blood was white and had no hemoglobin, and you know, it created a firestorm in both kind of thinking about the diversity of fishes as well as the evolutionary biology of fish. And if any of you are kind of history buffs and have studied the history of Antarctic exploration, you'll have to know of Sir Ernest Shackleton, who's basically had a failed expedition where they lost their ship and they were stuck on Elephant Island for years, but nobody died. And he says, this of course would say people wanted now, for hazardous journeys, small wages, bitter cold, long months in complete darkness, constant danger, safe return doubtful, honor and recognition in case of success. I think it kind of sounds like an ad for graduate school. But just to prove my point that the discovery of these fish added some heat to evolutionary biology, here's Louis Dollo, who coined Dollo's law. That is that once a lineage loses a complex trait, it can't re-evolve it, essentially. That's kind of a paraphrasing of Dollo's law. That's Gould's paraphrasing of it. But anyways, Louis Dollo monographed these Notothenioid fishes in 1904 and actually sketched a phylogeny for these guys, and was using this group of fish as an example for regressive evolution. And what I love about this is from Dollo's monograph. Basically, if you asked a fourth grader, maybe a third grader, draw me a map of Antarctica, they would draw something that looks like this. What I'm trying to point out here is that we knew a lot more about the biodiversity of Antarctica than we did the geography of Antarctica at this time. So really, the motivation for the exploration of Antarctica, of course, was to chart for geography. This is the last gasp of the European imperial age, right? This is 10 years before World War I. But what we see here is that much of this motivation in terms of let's get work done was really directed at describing the biodiversity. So I guess my point is, like, yeah, I may get lucky and catch a new species when I'm down there, but the point I'm trying to make is that we have a good handle on the biodiversity of these fish. And I'll just say that, now, like, the descriptions we're seeing now are kind of splitting up, more complex, closely related species, such as this group here, Pagonophreni. Okay, so really the one thing I'm going to say about biogeography is that we have this consistent pattern. That is, the species that live on the outside of the Antarctic tend not to be closely related to those that we find in the Antarctic. So here's another way of drawing that phylogeny. And all I've done here is red live in the red areas, green live in the green area. That is, the group of fishes of these Notothenioids we find in Antarctica are more closely related to each other than they are to these guys. So the fact that these Antarctic fishes had adaptations I will tell you about, we're really lucky because we have these species that never evolved the adaptations to deal with these polar conditions because they've never had to deal with these polar conditions. So we have a really nice, what we call a transitional series, where in a way, if you really want to think about it, these red lineages really are living fossils in a sense. They're giving us clues as to what Notothenioids looked like before they evolved features to deal with the polar climate of Antarctica. And I'll just make a note, this is more for Mike Bernard's interest, that something that Mike and I have talked a lot about is that in North America, freshwater fish species, sister species, that is, species that are most closely related to each other, are never co-occur. They're never what we call sympatric. However, we have this really interesting pattern in Antarctic Notothenioids that more than 80% of the species have a circumantarctic distribution, such as this species here, and only very few of the species are locally endemic to small areas of the Antarctic continent, such as this species here. Okay, so why do I think Notothenioids are in adaptive radiation? And I could be coy and say, well, because Professor Joe Eastman told me so. In his fantastic book, Antarctic Fish Biology, it was published in 1993. But I think we think about a definition of adaptive radiation, and this is my favorite, is basically adaptive radiation is when a lineage evolves ecological and phenotypic diversity and they're speciating rapidly, okay? And this was Dolph Schluter's definition that's paraphrased from a famous mammologist named George Gaylord Simpson, one of the authors of the Evolutionary Synthesis in the late 40s and 1950s. So if we look at Notothenioids, why do we think they're in adaptive radiation? Well, here's a snapshot of Notothenioid species at McMurdo Sound, which is the main US research station in the Antarctic, south of New Zealand. And what we find is that we have, all of these are actually quite closely related to each other relative to even other Antarctic Notothenioids. And what we see is a diversity of morphologies that are associated with the utilization of very specific habitats and resources. And one thing is that most teleost, or actually most ray fin fishes, regulate their buoyancy by having an air bladder, which is homologous to our lung. Basically, Our lung is actually the primitive condition. The air bladder is the derived condition. However, Notothenioids completely lack a swim bladder. However, even though they lack a swim bladder and they all should sink like a stone to the bottom of the ocean here, what we see is here is the percentage of body weight in water. So 0 means that they're neutrally buoyant. What we find is that there's quite a bit of variation in the buoyancy that these organisms attain, even though they lack a swim bladder. So what we're seeing here is a real strong phenotype-habitat correlate among these species. The phenotype being even general body shape, but also the phenotype being this characteristic of buoyancy. And there's some really neat stories about this. This guy is a silverfish, Antarctic silverfish, that's eaten by this guy. This is the sister species of the Chilean sea bass, which you guys may have eaten before. And what we find, both are neutrally buoyant, meaning that they have no weight in water. So if you anesthetize that fish and you put it here in the water column and let it go, it stays there. It doesn't sink, it doesn't float, it stays where it puts itself. It has neutral buoyancy. And both of these obviously are occupying non-benthic habitats, and what we see is that both have interesting but different adaptations for attaining neutral buoyancy, and they involve the evolution of unique lipid deposits. And what's kind of neat is that the silverfish eats krill, and the silverfish is then eaten by this guy here, which is called the Antarctic cod, or the Antarctic toothfish. And what we see, if we look at all these different species of Antarctic Notothenioids, and we look at their buoyancy, that is down here they're neutrally buoyant, here they're heavy, we have a lot of variation, but not much variance within species in terms of their buoyancy. And this is myself on a ship about to weigh a fish in cold seawater that's in that garbage can. So that's how the high-tech methods we use when we're down there to get this data. Okay, let's talk a little bit about this key innovation of the AFGP. And basically this picture tells it all. So if we were in the Antarctic and we saw this fish which lives on the underside of the ice, which we call the platelet ice, and we said, oh, I want to touch that fish. And we took your glove off your dry suit, which I don't know why you'd want to do that, and you touch that ice, immediately what's going to happen is because that water is super cooled, your hand is going to freeze solid. That ice crystals are going to whoop right to your hand, and you will have a frozen, damaged hand. So how can this fish rest on the surface of this ice without freezing solid? Now, this is what should happen to that fish. So if you take a temperate fish, this is a temperate surf perch off the coast of California, and you look at its freezing point depression before and after dialysis, what you see is that indeed there's some freezing point depression that's facilitated by sodium chloride and other small ions in its body fluids, but we're talking negative 1.86. That's clearly not going to be enough. However, if we take an Antarctic Notothenioid, such as this ice fish here, and we look at before and after dialysis, what we see is that there's this additive effect of these really small proteins called antifreeze glycoproteins, along with increased salts in the blood that give a freezing point depression up to negative 2.2. And even on its own, antifreeze is not enough. Antifreeze protein is not enough to avoid freezing at negative 1.86. So what's going on? Excuse me. Well, my mentor, Art de Vries, discovered in the 1960s that these critters make an antifreeze protein. It's a simple alanine, threonine repeat with a couple of sugars attached to it. And the way they think it works is that this is actually kind of theoretical physics here. They think that it disrupts the growing plane of ice crystals and just physically prevents ice crystals from growing. And what Art's wife, Christine Cheng, has shown is that the antifreeze glycoprotein actually evolved from a trypsinogen-like gene. And this is fast becoming a classic story in molecular evolution where the trypsinogen-like gene was modified and it made this antifreeze glycoprotein, and then gene copies of this antifreeze glycoprotein have diversified within these Notothenioids. It's not a single copy gene. Now, Notothenioids are not the only fish clever enough to have antifreeze proteins. Antifreeze proteins are quite diverse. And in fact, cods, which are kind of the dominant fish in Arctic habitats, have the exact same protein, but it's coded from completely different genes. So here's a really nice example of convergent evolution. The point is, these Antarctic Notothenioids have this antifreeze glycoprotein. They're able to avoid freezing. And those other non-Antarctic Notothenioids that are in southern South America and southern New Zealand, they do not. Okay, so with all that background, maybe I'll start talking to you about what I actually do, which is the phylogenetics. And as you can see, phylogenetics of Notothenioids is a pretty active field. A lot of people are making really interesting contributions. So what we're doing in my lab right now is we have about 80 of the 125 recognized species. And for a preliminary data set, we're sequencing a slug of mitochondrial nuclear genes. And we're actually getting preliminary data on 8 other single-copy nuclear genes. And we're exploring what are called species tree inferences. And basically the idea here is that state-of-the-art in phylogenetics is that you go, you collect a bunch of gene data, but when you do an analysis, you treat it as one gene. Now, species tree basically is, the real phylogeny here is the big tree in black. And we know that gene trees will not always match the species tree. That is why there are some genes that say that humans are more closely related to gorillas than they are to chimps. We know that's wrong. And the reason why that's wrong is because you can maintain polymorphism that you've inherited from your ancestors, and it just, by random chance alone, the way those alleles are sorted among lineages, you could essentially infer the wrong tree from any given gene tree, okay? So this is a whole body of methods called coalescent methods, a whole body of theory called coalescent theory. And this is just an example of this. Here is a group of Antarctic fishes called the ice fishes. These are the ones that lack hemoglobin. And so I went in and I sequenced the alpha-globin pseudogene. And what we find is that these 3 clusters, these 3 alleles, these are exact same alleles down to the nucleotide, are shared between all of these species in each of these red boxes. No one ever argued that either of the species in these boxes are closely related, okay? Why do they have the same allele? Because they inherited it from their ancestor. And the gene copies haven't sorted to match the species phylogeny. So we know ancestral, this is called ancestral polymorphism. We know it happens. Here's a case of it. In fact, we have alleles that we share with chimpanzees. Now that's not to say that, we're chimps. It just says the reason why we have the same allele at some loci is because our common ancestor had that allele. So if we look at these kind of exploratory 8 single-copy nuclear genes, we find some general trends. But now what we can do is we can kind of think about these new cutting-edge species tree methods, and we get a really interesting and resolved branching diagram that the punchline is indeed supports previous hypotheses that the Antarctic Notothenioids form a group. They're all more closely related to each other relative to these living fossil Notothenioids that live outside of the Antarctic. Now, however, to kind of go back to the old school, probably less precise way of doing this, we're doing a set of analyses we call Bayesian phylogenetics that uses an explicit model of molecular evolution. And we basically applied this to the combined data set of these nuclear and mitochondrial genes. And again, we get a branching diagram that we interpret as a phylogeny. Every point in the node, we call a node, is where lineages branch off, that has an asterisk, is essentially a node that's strongly supported. And indeed, what we find is that, again, the Antarctic Notothenioids form a group. Now, remember, we want to ask, can we take that phylogeny, draw it to absolute age estimates, and correlate it to the paleoclimatology and the timing, and can we just get at the timing of diversification of these Notothenioids? And then also, more specifically, does the timing correlate with periods of climate change that we see in the Antarctic that I've talked about? Both the fragmentation of Gondwana at about 80 million years, that part that involves Antarctica, and about 35 million years for the development of polar conditions. And this is compelled by this example with penguins. I mean, everybody loves penguins, right? And what they found was that at periods of climate change is where you had the radiation of penguins out of Antarctica to non-Antarctic areas such as South Africa, southern South America, New Zealand, all these areas that are not Antarctica where we find penguins. Okay, so I'm not going to read all this at you. Basically, this is for those of you who are philo nerds like myself. This gives you kind of the background of how we're going to actually estimate the divergence times. Now, the problem is rates of molecular evolution are not uniform in all lineages. So we have to take that rate variation into account. And we need some information, external information on absolute age. And what we can do is we can use that Notothenioid fossil I told you about. It's closely related to this guy to put a minimal age estimate on this node in the tree. And that's exactly what we did. So this is a little more background about that fossil, why we think it's a Notothenioid. And then here's some information about the actual geology of the formation, or the chronology here. So these oldest, what are called telms at this formation, that have been dated are about 51 million years. The earliest are 35 million years. It's good that the old ones are lower down and the young ones are up. That's nice. And this is based on strontium isotope ratios. And the telm that bears this fossil is here in telm 5, so it has a range of age between 47.5 to about 44.5 million years. So we could use that information to have a distribution, a probability distribution on the age for that node in the tree. So we know that the fossil is, minimally this old, has a peak probability of being this old, but 95% probability, cumulative probability of being that old. So that's a way of having a distribution where we're allowing kind of the error in thinking about the timing of that fossil. Oops, sorry. And sure enough, so now we're interested in asking, okay, we could calibrate what is the age of this Antarctic radiation? What's the age of the entire Notothenioid radiation? So what I'm showing you are the ages inferred from each of these different genes. And what we see is that we get a range of ages from 55 million years to about 80 million years. And here represents what we call the 95% credibility or highest posterior density. So that, in a sense, is a way to think of it as a credibility interval. Definitely not a confidence interval, or a Bayesian statistician will freak out if you say that, because this is a Bayesian analysis. And then here, this represents the molecular evolutionary rate for each of these genes. Now, this is the age for the Antarctic clade. And remember, Antarctica, we think, became cold about 35 million years ago. And we see this rather compelling convergence of age estimates all right at about 30 million years. And indeed, if we think about the credibility interval here, we're bounding that time that we think Antarctica really started to get cold. So what we're seeing here is that the timing of the radiation of this group correlates rather nicely with the time that we observe climate change in the Antarctic. And now we could kind of combine all those genes into one analysis, and that's basically what I've done here. And the branches here correspond to geologic age, geologic time here in millions of years, and the gray bars represent those credibility intervals on each of those age estimates. And what we see is that the entire Notothenioid radiation is bounded right about that 80 million year time where Antarctica is fragmenting from those other Gondwana landmasses for which these living fossils live. These living fossils are found in those landmasses that Antarctica used to be connected to. And what we see here is an age that sits right on the timing where we see this rapid precipitous decline in temperature, not only across the world, but in particular in Antarctica. So we can ask, are there correlates of Notothenioid diversification with regard to divergence times with paleogeography here and climate change in the Southern Ocean? And I would argue indeed, at about 80 million years, we see the diversification of Notothenioids, the initial diversification, and this is coincident with the fragmentation of East Gondwana. And so indeed, there's a possible role for fragmentation of continents separating groups. And this is called vicariance. When you have the formation of a barrier to dispersal via some kind of abiotic factor, that's what we call vicariance. And then we see at 30 million years, we're converging on an age estimate for the diversification of those Antarctic Notothenioids. And indeed, this is coincident with the formation of Antarctic ice sheets that we hypothesize occurred at the Eocene, Oligocene period, where we see a pattern of global climate change. So now we can think about these Notothenioids in a really interesting light. all the stories of adaptive radiation. I had lunch with the graduate students today, and one student rather intuitively said, so what you're telling me is that Antarctica is kind of like an island. And I said, well, yeah, and this student was wise enough to know that most of adaptive radiations are studied in islands or isolated parts of the world, such as Rift Lake cichlids. And if we look at kind of some of the canonical adaptive radiations reviewed here by Solly Seehausen, what we see here, approximate age in millions of years, essentially all adaptive radiations are really, really young compared to these Antarctic Notothenioids. So now we could put the context of the biodiversity we see in the Antarctic in terms of the phenotype-habitat correlation, what these things are doing to make a living, how they've diversified ecologically, morphologically. We could put the context of the evolution of novelties, such as these antifreeze glycoproteins, which you have to be swimming around in this habitat, let alone hanging out on the ice like this guy does. And now we could put all of that into a temporal context. So now we could really develop interesting hypotheses about not just rates that these lineages are diversifying, and by the way, these guys are diversifying much faster than these living fossils do. There's a significant shift in the diversification rate here. But we could really think about the rate that characteristics such as changes in habitat utilization, the phenotypes that are associated with the utilization of those habitats, really put that into a broad temporal perspective. So in conclusion, I would just like to, again, thank everybody for inviting me to come share my work on these Antarctic fishes, and particular people in my lab, Kristen Kuhn, a postdoc, undergraduate researcher Jillian Pennington, and two of my lab techs have contributed a lot to this work. Christopher Jones is kind of my meal ticket down to Antarctica every 3 years, and part of this work was funded by NSF, and I'll close with a shot of an Antarctic sunset. Thank you.
Does anyone have any questions?
Oh, all right. I feel like it's Oprah or something.
That's right.
Tom, I think for people arguing about sympatric speciation, some of these, the cichlids and then the sticklebacks have been one argument where you have adaptive radiation diversifying selection tying into sympatric speciation, and you sort of floated out Barraclough and Vogler's method with the distributions. I mean, do you see any signal in the distributions of sympatric speciation in any of these fishes?
Yeah, I mean, that's a really good question. So the way we think about sympatric speciation is we need two things. We need things living close together, and you're not going to believe this, Mike, but I have slides from the talk that we co-authored at Evolution that's germane to this. At least I think I do. No, I don't. Okay, so we need two things. We need lineages, closely related things that live close, that are in the same place, and we need some signal that they're different morphologically, or they're different in utilization of usually discrete resources or habitats. And the model is that basically what you want to see, if you went out and measured the characters, or you measured what they're eating or where they're living, you're going to see a bimodal distribution. And what we see for this guy right here, this is a group, Paganophryne, 17 species, essentially all of them are sympatric. A study was done, and they're all very closely related genetically, like they've diversified within the last 2 million years. Several studies now, 3, have gone in, caught several of these species in the same trawl, and basically they're all eating the same thing, and they have the same kind of mouth. They've got this interesting barbel that they think uses a lure to fish in large macroinvertebrates. And the only thing that's really different are kind of pigmentation patterns. So we're not seeing that bimodal distribution among closely related, really sister species of either morphologies or resource utilizations. So what I think is going on, this is kind of my pet hypothesis, is that what we're seeing is a ghost of allopatric speciation past. And what we know is that, one part of the story I didn't tell you, is that during the Pliocene and in the Pleistocene, so roughly from 5 million years to the present, there have been periods of global warming in the Southern Ocean. And in fact, there's evidence of large outflows, freshwater outflows from the Antarctic continent to the Southern Ocean. So these fish, if you take them to 4 degrees centigrade, they die. That's too hot for them. For us, you know, it's something like 42, right? That's where we start cooking our proteins. Their heat shock proteins start kicking in at about 4 degrees centigrade. So these warm water outflows could have been barriers to dispersal. Not only that, they're marine teleosts. They don't like fresh water. So even though Antarctica is rather homogeneous today, it may not have always been homogeneous. In fact, there's Pliocene age Notothenioid wood. Notothenioid is a southern beech tree, and it's a kind of a classic Gondwana group of plants. There's wood that dates to the Pliocene on the Antarctic continent. So near shore, there might have been some near shore habitats that became a little more temperate. So I think of these as kind of providing barriers to dispersal around the continent. And you could have had allopatric diversification, then as Antarctica became more homogeneous again, facilitated, you know, co-occurrence. Yeah, gentlemen, or?
I think Roy had his hand up next.
So I'm just curious, how rapid do you think that the change happened when this broke through? I mean, was it one day a waterfall broke through and it all of a sudden started coming in, or were there marshes and things? And do you have to have a sudden change, or is it something that over the course of 1 million years things started to change a little bit? And what kind of latency would you anticipate before you start seeing this sort of?
You mean the signal of the correlation between the timing of some paleogeography?
Yeah,
and
when you finally get this circulation,
right.
would you anticipate, you know, that it would be 10 million years?
Right. I think it's probably smaller. And
would it make a difference whether the change was sudden or?
I don't think it would, because I think ultimately with you think about, you know, development of freezing conditions, whether it's over the course of, you know, a few million years or 10 million years, that ultimately you're going to get this extreme change in the habitat. And not only that, it's not just the freezing conditions that really hurt these fish, that most marine fishes are found in nearshore continental shelf habitats called the littoral zone. And essentially there's very little continental shelf habitat in Antarctica for 2 reasons. One, there's so much ice on the continent that it's pushed the continental shelf, the slope up. And second, there's constant glacial scouring. So, you know, you're hanging out, you know, you're a happy community, and then this glacier comes and basically obliterates those habitats. So, you know, maybe in theory, if it happened over a longer period of time, would have facilitated adaptation in other lineages. I don't know. And maybe if indeed it was perhaps more sudden than, you know, we can kind of wrap our head around thinking about that timing, then maybe it just kind of, you know, facilitated at least, you know, the sense only one group evolved these adaptations. I don't know. It's an interesting thought problem, though. But there are consequences of the timing of that. One of the canonical biogeographic marine barriers is the formation of the Isthmus of Panama about 5 million years ago. Well, actually, they think it's, well, anywhere between 5 and 2.5. And depending on so marine fish species have been isolated on either side of the isthmus. Those are called geminant species. And if you look at the timing of those splits, and you even take into account the polymorphism in genes like I was talking about, what you find is a wide range of divergence times. And what it was thought is like, no, it wasn't immediate barrier, but it was a gradual barrier. So those fish species that were more kind of marsh, brackish water adapted were still dispersing between the Atlantic and the Pacific, whereas perhaps those that were more reefy kind of fishes, the barrier was more immediate.
This isn't quite what you're studying, but do you know if anybody knows with regard to those antifreeze glycoproteins, whether the major antifreeze activity is due to the protein component or the carbohydrate component?
Or the sugar, yeah, that's a good question. I'm not sure, actually. I don't know. Yeah, no, it's a really good question. It's the sugar? Okay.
Along those same lines, you mentioned that there are some Arctic fish that have
Right.
antifreeze glycoprotein. I think there are some people who believe that the glycosyltransferases that attach the first sugar to the protein are very highly specific with regard to their substrate. And I wonder if anybody's compared the glycosyltransferases as a way of looking at this convergent evolution between the two different.
I'm not familiar with that. I'm not sure. Do you know, have some insight on that?
All those groups that study the Arctic fishes are also doing that.
Okay. And they're comparing to the Antarctic fishes, I see. But it's clear it's convergent. I mean, these are, if you look at the evolutionary age of the common ancestor between the Arctic and Antarctic fishes, you're talking about 150, 100 to 200 million years. So it's a really deep split in that fish tree of life.
One more if I could.
Yeah, please.
Is it the same protein that was adapted for the same protein? In other words,
They
the
don't think it
trypsin-
is. Now that part of the story I've been given some insight on, and I think that they've been able to rule out that it's an origin from a trypsinogen-like digestive enzyme. So.
Any more questions?
Darren.
What are the rays doing down there? They're the other clade that.
Oh yeah. Well, they've got urea in their blood, so that's what they do. They have kind of a different strategy for osmoregulation, and I'm sure Ron would go on. I'm sure he'd teach this in your ichthyology course, right? How sharks have different osmoregulation, right? And because of that, as a consequence, because they have more salts in their blood, in their body fluids, they're able to avoid freezing.
But are they mostly pelagic? Because you had there was a lot of diversity of
Yeah.
chondrichthyans that.
Yeah, no, right. So no, they're all benthic now. It's one genus called Bathyrraha are the ones that you get down there, and there's probably 7 species or so. But they're benthic near shore. They're pretty abundant too. Not abundant enough to upset the Notothenioids as being the kings of the Antarctic oceans, but yeah. Yeah, Joe.
I'd like just to return to the issue of sympatric speciation versus
Sure.
the echo of allopatric speciation.
Right.
So if you do look at some of the species flocks, like the cyprinid flock in Lake Tana, that is almost all ploidy differences. So in terms of, I don't know, not knowing this group of fishes, I mean, are there ploidy differences, or in those situations where you are sampling very closely related species in the same general location, is there anything known about the behavioral differences or the life cycle differences or spawning habitat preferences that are different for those?
Yeah, there's very little known about that. What we know is that at least that one non-Antarctic, what I call living fossil, that is that we think the fossil itself is related to, that is thought to be a spawner. Then what we think is that the entire Antarctic clade are probably ancestrally have male parental care, nest guarding. So we've seen it in several of the major clades of these Antarctic Notothenioids. However, the ones that are pelagic, that little slide I showed you of that little kind of, that kind of food chain there, the krill to the silverfish to the Antarctic cod, those two species are thought to be pelagic spawners as well. In fact, the silverfish now they think is spawning in the platelet ice, that underside of the ice that other fish was sitting on. So given that now what we've seen, and this is only through divers and actually luckily pulling up a nest and a trawl, so given that if there's strong male parental care, that opens up, I think, the avenue for sexual selection to be at work. And what we see is that you will find subtle differences in terms of there is dimorphism between males and females, both in body proportions and pigmentation patterns, but nothing like brilliant colorations like we see in cichlid fishes or darters here in North America. So I think that whole avenue needs to be explored in terms of the role of reproductive strategies and thinking about speciation. And it's just been so hard that they're really hard animals to get going in the lab. Even the small species have generation times that are on average, 7, 8, 9, 10, 12 years. So it's been kind of, it seems like all the cards are stacked against doing interesting experimental work in those areas.
What about the issue of ploidy differences? Are there
No, but there are differences in the chromosomal complements, but they're all diploid. But what I find actually really interesting is that the groups of fish I work on, which are, other than these guys, are darters and sunfish and black bass, which are both endemic to North America. There's a darter there. Essentially, you know, through long stretches of evolutionary time that we think of these clades been around, they essentially have the same number of chromosomes. Whereas there's a lot of variation in the chromosomal number in these Notothenioids. So that there might be something there. And I think the scale that people have been doing this work has been more like, well, let's fill in the big picture clades, and no one's really gone into kind of those little species flocks and asked, are they different? Because that would be an immediate kind of post-zygotic mechanism potentially. You know, talk about a Jabjanski-Mueller incompatibility. If you have different number of chromosomes, that's probably gonna work to prevent kind of as a nice post-zygotic mechanism.
Joe, I don't know anything about Antarctic fishes, but my feeling just from a couple pieces of data is probably that the polyploidy issue with cyprinids is probably something that is, my guess, endemic within cyprinids, because that same pattern that you described in the African cyprinids, that occurs in other cyprinid lineages also, and that also polyploid is associated with the evolution of catastomids from a cyprinid ancestor.
Right.
So cichlid fishes, which I study, are extremely diverse, and they've been karyotyped like crazy, and there's no evidence for any kind of polyploidy at all. So here we've got a couple examples of at least no clear examples of polyploidy in all these other lineages that we're talking about, but multiple examples in one lineage that you brought up. So that leads me to believe that it's probably something specific to that lineage of fishes that
Right.
allows them to undergo polyploidy.
That's right. And you know, there's actually, to speak to why do we have these sympatric species and what are they doing differently, one thing it could be, not to belabor this point, is something similar to the whitefishes in the Great Lakes, which are no longer with us, where you had all these very similar species that co-occurred. Like you could go off the coast of Lake Michigan, Chicago, go fishing, catch 5 species, and you're like, what are these guys doing, you know? Well, what they were doing is that they were spawning at different parts of Lake Michigan. Like one species would go to Green Bay. They were Packers fans. Other species would spawn off the coast of northern Indiana before they built all the steel mills. And so there was this spatial isolation in terms of spawning. Then there was temporal isolation, that different species would spawn on these gravel shoals at different times of the year. So that could certainly be going on as well among these closely related species. And we just don't really know. The natural history isn't there yet. No.
Are there any other questions? Okay. All right. Thank you very much, Dr.
Thank you.